Hi, I am Ben Gutierrez. I am the curator for the apple and tart cherry collection with the Plant Genetics Resources Unit in Geneva, New York. And today we're just going to kind of explore the genetic diversity that we have. It's really important to understand that we've got the best job in the world, right? Like this is some beautiful diversity that is just at our fingertips, and as we are the stewards, right, or the guardians of these wonderful collections. What a tremendous job and responsibility that is. And thank you so much for participating in the poll today. It's interesting to look at it. We know what our priorities are, right? Peter Bredding has taught us well and coached us on that. But sometimes our challenges aren't necessarily what our priorities are. I know for me, distribution is a pretty significant challenge when we look at all the non-research requests, or like you see some of our crops get very little attention, and how do you promote those? And so sometimes, it's interesting to take a step back, and especially I think for perennial crops where we have the seasonality to our work, and this seasonal availability, or the maintenance challenges change throughout the year. And it's kind of fascinating to see the commonalities that we have, and the unique struggles that each collection has. So we should be so proud of our work, and I love looking at the holdings each year. As we see the numbers increase, we're holding more accessions than ever, and we distribute nearly a quarter million. Like that's so exciting that we really are impacting the world and making that germplasm available. And I love this quote, and I always share it when I speak to groups, that it's really our job is so important for sustainability in agriculture. So diversity is a way of coping with the possible. It acts as a kind of insurance for the future, and that's really our role in American agriculture. Here we are in Geneva, and a very distinctive climate from our friends in Corvallis and Davis, Riverside and Parlier. But that's by design, right? Like we all have unique crops and unique climates that we manage. Just briefly, our PGRU operations, a little bit unique because we do have a field team that's supported by any 9 funds managed through Cornell University. So our farm manager is a Cornell employee. We have several field technicians that are also Cornell employees, and we really couldn't function without them. And it's a wonderful resource that we have for our collections. Recent managerial changes, now the field team manages all crops, clonal and vegetable, and that's given us some growing pains, but it's been a wonderful process. And I think having a more united field team has been a benefit to us. Everyone knows Dawn. Yeah, she's our program support assistant. And very exciting, we have a new industrial hemp collection with a new curator. And so lots of exciting changes happening in Geneva. My old position as Vitus curator is now coming out, so we're interviewing soon, starting next week. And many of you probably knew Heidi Schwaninger. She passed away in late 2018, and now we're sad to have lost her, but we're excited to announce that we're hiring a new molecular biologist to take her place starting on Monday next week. Wonderful interns. I mean, so interns, are such a resource for our collection to evaluate, to come in and actually provide that training and enthusiasm for what we do to the next generation has been inspiring. We had an incident in our greenhouses, and we lost, our greenhouse and also our lab facilities, and really our hub of our operations due to this fire. And no germplasm was lost, which was fantastic. But it really did set us back operationally. But now we have made do, and we are fully operational as of 2021. Again, it's just looking at our footprint. So this is from 2014 to 2018, and just looking at all MPGS sites and our footprint throughout the world. It's kind of remarkable to take a step back and look at all the places that we've touched. So this is just Geneva, which includes our vegetable crops and our clonal crops, and just seeing how far a net that we're able to cast, right, to make sure that we're delivering the germplasm, which I think is such an important priority for our work. We've recently been looking a lot at our apple collection and looking at the impact that we've had since its beginnings in 1984. So this is looking at apple distribution throughout our history. And, going back to 1988, where we have our first GRIN record. So really this would have been the collection started in 84. And now the collection's finally big enough to really make a material available and start distributing the germplasm to people. I grouped the requests by their affiliation. So we have a lot of unaffiliated requests throughout our history. And this number is really only is been growing. And it's kind of interesting to look at the pattern of the research requests. Some years were very heavy, but now it's, steady increasing of our hobbyists and other just unaffiliated groups. And we're trying to look at how to better manage that, because I know that it does increase the burden on our field team, and we do want to make sure that the germplasm is being used appropriately. This is the general view of what people think when they hear about gene banks. They think of security. This is Svalbard, and they think of material that's safely locked away and preserved for doomsday. Right? This is the doomsday vault. And but this is our view of gene banks. So like the clonal repositories are so unique because they do balance this vulnerability and accessibility. And so here is our collection, our apple collection on full display, right? Full access for research, for collecting material at any stage, but it's also incredibly vulnerable to biotic and abiotic stress, which provides some unique challenges. So it really is, we're trying to balance the how accessible is our collection versus how vulnerable is our collection. We do have one of the largest apple collections in the world, with about 56 species and hybrids and close to 6,000 unique accessions. The grape collection we do share with the Davis repository, and that we manage the cold hardy grapes with about 30 species and hybrids and of about 1,400 accessions. Our tart cherry, our prunus collection is quite small with only 135, focusing on the tart cherry, but we do have some wild relatives of that crop as well, and they are cold hardy, and they do well in our Geneva environment. So this is looking more closely at our distribution over the last 5 years, and we do see apple is king, right? We have such a tremendous resource and very unique collection that attracts a lot of stakeholders, and we get so many requests for our apple and relatively few for our grape and our tart cherry, which are really get focused on by a unique set of stakeholders. This 2021 is interesting because it was not due to COVID policies that restricted distribution, but it was due to fire blight. So this is the vulnerability part of clonal crop preservation, is that we get diseases, and sometimes they blow right through our collections, and we had a terrible outbreak of a streptomycin resistant strain, which really it didn't respond to our chemical treatments, and by the time we identified the strain it was much too late, and we had to do a lot of season pruning and some chemical mitigation to manage it. We're doing much better now, but this was the look that we saw last June and July, kind of very discouraging to see. This was a fascinating picture that we grabbed. So this was two accessions side by side. One was maybe 50 to 60+% fire blight incidence, and here's one that very nice looking clean genotype, clean accession with fire blight on the root suckers. So here, we get to look at this contrasting, vulnerability to a disease pressure, where we have potentially some nice disease resistance from our apple collection come out. And so, there's a timelessness to what we do. So some of these cultivars that we preserve are so old and so important to the modern breeding programs today, and it really is this cool, you get to look at the history of your crop at the same time as well. It's such a unique view that we're able to enjoy. So really, as a new curator, relatively, we're trying to really understand the process, and as we've changed our management program to include the vegetable crop maintenance, we do want to make sure we know where we are at any given point, right? We know what the priorities should be for our crops. So we're just trying to really map out the processes, and understand, what are the priorities and what are the unique, challenges for each part of the season. So, we do have the benefit of dormancy. So from really December through March, it's not quiet. It's never quiet here, but we get to focus on the pruning. Our field team does all of the pruning now. Our distribution, so it is, I think, a little bit more leisure than in the warmer climate collections, but we do have some unique challenges to manage because of our warm, wet New York springs and summers with disease pressure. The we talk about priorities and challenges. So like the repropagation plan seems simple, but it's an ongoing challenge to manage not only the plants in the nursery, but their process in the cycle, where they are, what's the next step for them, and just keeping your sanity as you follow these things through. It's really a 3-year cycle from making a new tree through grafting and then bringing it throughout the whole cycle and planting it on the 3rd season in the summer. So it's it's important to understand when we have new opportunities to bring in accessions. So we have summer budding that we're able to do for for apple and tart cherry, and we can bring in new accessions there. And our in December we have our dormant scions that we can bring in our cryopreserved budwood, and also any new accessions that we bring in. But again, it's keeping track of inventory, because there are some, even the nursery isn't safe, so we need to follow up on the nursery constantly to make sure that the trees that we've are repropagating haven't been lost in the nursery as well. We need to start the process over. Grape is a little bit simpler because we do not graft. They are self-rooted, and so it's a 1-year cycle, and we're able to get a new vine planted in the permanent collection from a cutting. Here's Todd Holleran from the Apple Rootstock program. His hands were famous in the Finger Lakes Times from an outreach event that we did. But it's grafting is really the simplest way to propagate apples and tart cherries, and it's an ancient technology that is still so useful and valuable for propagating this timeless germplasm. But here's kind of just the rough process that we go through. This is the benefit to our unique set of Vitis materials, that they don't need to be grafted onto rootstocks, and in fact, having them self-rooted is a benefit to us, because we can still rescue the genotypes even if the above ground part is damaged from disease or from any number of things really, but cold hardy damage as well. Cryopreservation. This is a big shout out to Gail and the team in Fort Collins, because I do think that cryopreservation and germplasm management gets this kind of this is a doomsday collection, right? This is for catastrophic events, but really we rely on cryopreservation for really our routine maintenance as well. So we lose things every year, it seems. The same accessions we know are troublemakers or are going to get disease, and we rely on the cryopreservation programs to rescue those lost accessions. And really, without this, we really would have a hard time keeping up with the repropagation cycles, and we would constantly be repropagating large portions of our collections that we couldn't rescue from cryo. Unfortunately, and excitingly though, I think, is that there are challenges to the grape cryopreservation, not as simple as apple or tart cherry maybe, but I think the team has some exciting new techniques that they can use, the droplet vitrification to cryopreserve grape, and that is going to be huge for the grape collections in Davis and in Geneva. I love this because it's what's in a name, and this is one of our challenges, right? We have these collections, and so this is Malus prunifolia, and you can go and look at all the different accessions and see tremendous fruit quality variation and morphological characteristics. So begs the question, which one is right? Which one is the true to type and representative of that unique taxa? And we don't know always. So a lot of the focus tends to be on the domesticated crop, and so little is known about some of our wild, crop wild relatives that we are managed and that are so important to breeding and for the research. But really, understanding this puzzle helps us. So when people make discoveries using a Malus prunifolia accession, we know the biological and botanical relevance of their work, so that people aren't maybe pursuing this material, they're pursuing something more true to type, which we're suspecting is maybe something more similar to this apple accession there. Vitis hybrids, we have most of our collection are called Vitis hybrids, which could be really anything. Typically, we think of vinifera, cultivated grape crossed by a wild grape. But really, hybrid is kind of a meaningless term to mean anything interspecific. And so we can look at our grape collection as hybrids, but that name doesn't really pull out much information, it by itself. So we can look at the percentage of its vinifera background. And so here's a stout seedless hybrid, right, that has 90% vinifera, so very different genetic makeup than this NAPCA accession, which only has 10% vinifera. And so this, we're really trying to work on the reclassification of our Vitis hybrid collection to make the nomenclature more meaningful to our stakeholders, so that they can, see a name or, search for accessions of a particular class and pull out that list for them. An apple a day. We always like to joke that we have, based on our field collection, we could have an apple a day for 15 years. And, as try as we might, I don't think I've still tasted everything. There's so many, the timing and the seasonality of everything is challenging to capture all of that diversity in eating a single season. But, we try our best every year just to get that qualitative evaluation. Apples are big worldwide. So USA is number 2 with 42 million tons. This is probably a little out of date, but so Washington and New York State are important sites for apple production in the world. Apple domestication. So this is a fascinating story that involves some interspecific hybridization, and it's not as not as dramatic as some of our other crops and their domestication and the events, but we were pulling out a lot of information from the collection, and it's been used quite extensively to look at the domestication process using genomics. And so we know that apple is a interspecific hybrid of Malus seversii from Kazakhstan and Malus sylvestris from Europe. And that process of grafting and trading germplasm throughout the Silk Route and introducing new traits and new species along the way has been so fascinating and enlightening as we look at apples, in a new light, right? As we seek to understand their history, we understand more, the possibilities for the future. But interestingly about apples, so this top row is our domesticated apple. This middle row are Malus seversii, which when you look at them, it's really hard to make that distinction of the two, different groups. So really, it's not quite as dramatic as some of the crops and their domestication cycles, where the domesticated form looks so distinct from the wild relative. This is Malus sylvestris, a little bit smaller, very acidic fruit relative to Malus seversii, and these are some of our North American apples just by comparison. And so some of the genomic studies that have been done using our germplasm have been really fascinating, because we do say, this is domesticated apples are a hybrid of 2 species. Well, there's the question of how much, right? And is it true for across the board? So we do see some interesting, diversity for the contribution of each of the progenitor species, and then the direction of the cross as well. So we do see mitochondrial or chloroplast inheritance switch a little bit. So we tend to think of sylvestris as the maternal parent, right? So we get that plastid inheritance through the maternal parent, but then there are some that don't have that, and they have a unique origin for chloroplast, or they share more with the Malus severusii, plastid genome. So it's kind of interesting, just it's a complex story involving, trade and time, and obviously, new breeding programs introduce additional Malus species, which makes the story even more complex today. And it's, again, our resources in Geneva have been tremendous for enlightening this process in domestication in apple. Looking at genetic markers, we have a lot of clones in our clonal material. So a lot of sport mutations that have been collected and propagated are, really exciting sometimes, but you do question as a curator, like, do we need this whole collection of red delicious clones, right? Like it's one genotype, theoretically, right, with just a few point mutations that distinguish the phenotypes. They're redundant, but there's use to them. We can also look at first degree relationships, which are parent, offspring, or sibling relationships. And we see like this beautiful interconnectivity. So there's a lot of allelic redundancy. But again, the challenge with clonal crops is that the unique allelic combinations are important. So we do try to keep siblings because those unique pairings of alleles are irreplaceable. And so as we move forward in the collection and start hitting some walls, right, with space and other management, it does come down to these guys, right, or reducing some of the redundancy or keeping some in cryopreservation and not in the field. And some of those hard decisions need to be made at some point. Here's like an example of russet mutations in golden delicious. So all the same genotype, but very different outward phenotypes. This is one of my favorite razor golden delicious, but it's exciting because we can evaluate it from a new perspective, right? So this is looking at floridsin, my chemical of choice, looking at apple, what I studied for my PhD, just a unique phenolic found in apple tissues. But we look at the same genotype, but now in expressing russeting, we see a strong fourfold increase of floridsin in fruit, which is kind of interesting and maybe a unique way for people to dissect that trait in the collection. So here's fruit development in golden delicious over time. We see increasing russeting, increasing nutritional content, right, potentially. And so so we talk about the sports are redundant, but they are useful for a lot of our stakeholders to evaluate some key traits. Explorations, so hopefully Gail will share some of her adventures in Vietnam. I wish Thomas were here to share some of them as well. So Thomas and Gail took this exploration and brought back a really unique species, Malus dumeri, found in Asia. And really, there was only a single accession to represent that in that species in the U.S. And now we have, so much genetic diversity with plans to go back and collect more. So this is a really, really unique tropical apple that our breeder at Cornell, Susan Brown, says that you can introduce cold hardiness in a single generation, which is really exciting. So useful traits found in this species could be brought in pretty easily, without getting that cold hardiness aspect. This is more data from our fire blight outbreak in 2020. So, really broad distribution in disease incidence, some up to 85%. This was really challenging. So it seems, kind of frustrating to watch your collection really melt away, but a great opportunity to collect some data. So while our field team and so many of us were busy pruning, we had a postdoc and field intern collecting data on nearly the entire collection. And here's kind of the rough distribution of disease incidence. So we can look at Malus domestica, the cultivated apple, and see, some had very little incidence all the way down to very heavy incidence. So it's like kind of a, lemons into lemonade opportunity to look at disease resistance in our collection. So then we've traced progression into July and saw a lot of these hardy genotypes are now becoming more and more susceptible to disease pressure. So because of maintenance, right, like we had to cut out the disease before it could progress too far, we also scored cut scores. So here's a tree that was very overcut, right? We're training our field team to not be this aggressive when you cut out fire blight, because we do want material to propagate to rescue that genotype. So a lot of learning on our part, and but we can look at diversity and disease resistance from a cut perspective across additional apple species. And so this is kind of a wonderful study to pair with our historic data, because some of our historic data, we find that in GRIN, it says it's very resistant, but last year we observed 55% fire blight incidence, or we do see some very susceptible but minimal incidents, and then some, some agreement with the GRIN data. But when we looked at it, GRIN incidents versus what we scored in 2020, which there could be some disease, the strain specificity, and things like that modify, right, the disease resistance in a particular genotype. But now it's just additional data that can serve our breeders or our pathologists who can take a more in-depth look at the disease process in apple. Tart cherry, I'm falling in love with this crop. It's kind of was orphaned for a long time because it is only 135 plus plus or minus accessions, but it's this exciting hybrid between our sweet cherry and the European dwarf cherry, Prunus fruticosa. And here's again where we have some overlap with the Davis repository in managing Prunus, but these are supposedly a lot more cold hardy and can do fine in our New York conditions. So we've been looking a lot at the bloom variation. So I say in question mark, right? Bloom variation, like they nearly all bloom at the same time. And this is one of the challenges for the tart cherry and really the cherry industry is like, how do you get these things to bloom later, right? There's very little phenotypic variation, at least in our collection, right? And so we've been also looking at different nutritional qualities, anthocyanins, melatonin, trying to better explore the collection that way. So when we look at the U.S. industry for tart cherries, it's really based on one accession, this Montmorency cultivar, about a 400-year-old French cultivar. And really, so when we look at the full diversity of our fruit quality that we've been assaying over the last 5 years, again, this is great intern effort in collecting fruit and evaluating them. We see Montmorency is the vertical line, and you see like it tends to fall pretty far from the potential except for weight, right? Like Montmorency is an exceptional yielding cultivar, not only like adequate flowers, but also beautiful fruit quality and or weight really. But like in terms of soluble solids, we could do much better. Tart cherry juice is sold primarily for its nutritional benefit. There is so much more chemical diversity that could be exploited in the tart cherry genetic resources. So again, it's like pushing an elephant up the stairs, right? We hope to do more with the collection, but getting the industry excited about new cultivars is the biggest challenge. Grapes, so I am a little bit sad to have left the world of grapes, right? Even though I'm still the acting curator, but this is the world's most valuable fruit crop and such a beautiful, array of genetic diversity and phenotypes and quality. And so we do spend a lot of our world in this Vitis labresca, Vitis hybrids, and these wine grapes, right? And so Davis has a wonderful collection of the Vitis vinifera and other key wild grape species. And this is cold hardiness, right? This is where, cold hardy physiologists, this is where they begin their day and their work. And the vines tolerate it. And because we do get some cane damage, but because they're self-rooted, a lot of the times, damaged vines will just regrow by their roots, which is, a convenient way to capture that repropagation. So we've been looking at phenological variation in our collection across several years. And this is a really important trait for climate control or climate adaptation, right? So, New York growing conditions are very distinct from Davis and from other grape growing regions. And we want to kind of like understand that phenological diversity that could be exploited. So one of the most critical stages is just bud burst. Sometimes the volatile New York or cold climate winter springs, right? They come out. We might have a 70-degree day in March that gets everything activated, and then followed by some spring frosts that leave things pretty damaged. So understanding how to delay bud bursts is something that one of our local physiologists is very passionate about. But we were just looking at the phenological diversity from our genetic diversity perspective. And you can see, very minimal diversity for bud break. So this is just a matter of a few days to a few weeks potentially, but that could, likely save your production if you were able to integrate some of the latest bud break genotypes, right? So more exciting is the bloom variation, and this is where we get really challenged because we're trying to distribute pollen all over the country. And so someone might request, this Ampelopsis accession, right, blooms really late, and they're hoping, because they're in California, right, they want it earlier. So there are some challenges to getting some of these late bloomers. And so for us, this data is really valuable because we know roughly when to go out based on the growing degree days and collect pollen for our stakeholders. It makes it much easier. And then veraison, right, that point at which the berries start to change color as a sign of maturity. But what's exciting is that we can take that genetic diversity and some of those traits. And I mentioned it at the beginning, right, like what's in a name, and hybrids, right, like that doesn't really capture what the full genetic diversity, and it doesn't represent a unique taxon because there's so much. So these accessions here are in gray are Vitis hybrids. And what we're able to do is look at the ancestry estimates compared to Vitis vinifera in black and wild Vitis in red. So here's a cluster of Vitis labrusca and other North American Vitis species. So what you do is you plot this using principal components analysis, the Vitis vinifera against the wild Vitis, and then you can project onto this plot your Vitis hybrids. And then you can estimate the ancestry by just looking at the Euclidean difference, right? So here's a particular accession. You can estimate the percentage of vinifera by looking at its distance from vinifera against its distance from the wild species, and you can come up with a percentage. And which we've done that, and we estimated and actually looked at the historical literature or pedigrees and expected percentages of vinifera, and actually came up with some pretty strong numbers for that. So now we have a way to classify our Vitis hybrids. So actually this cluster is really important because these are the Vitis labrusca hybrids. So Concord sits about here in the middle range. So it's about, anywhere from 40% vinifera according to the pedigrees, or our pedigrees, that is. But what we can do with that information is now okay, we have a classification. You can look at 10% hybrids against 100% hybrids. And when we consider growing degrees specifically, we can look at other traits as well. But when we look at, now looking at the group of Vitis against their growing degree days for bloom, we see this increasing trend with increasing Vitis vinifera. So this is really a valuable way to leverage our genetic data to help our stakeholders better utilize the genetic resources for our Vitis hybrids. I know that's been a big concern, is how do we distinguish, right? So this group down here are also called Vitis hybrids, right? But they are so much more closely related to the wild Vitis that, because you can have wild by wild hybrids or wild by cultivated hybrids, and any number of back crosses back to vinifera. Even some of these were misidentified Vitis hybrids, which actually are more 100% vinifera. So it gives us a chance to update our taxonomy using the genetic data that we have. I kind of ran through this because I knew we were starting a little bit late, but just really acknowledge the wonderful team that we have, the PGRU Plant Genetics Resources Unit, and also the Grape Genetics Research Unit. Cornell University are some of our top supporters, just because we're in their backyard, and our interns. Can't forget the wonderful undergrads that serve our program so well. And at that point, we have plenty of time for questions. I'd love to hear any feedback or comments that you may have.
Hi, Ben. First, I have a good comment, and that is I was able to reboot and I was able to start recording. So there'll be a first part if you could re-record some of your first slides, but hopefully we did capture most of your talk just then. Let me open it up for questions. I know I have a few, but I want to hear what some other folks have for questions. So go ahead, open mic here.
Hi, this is Jill. I take care of the Rubus and Ribes collections in Corvallis, and we have quite a bit of redundancy in our species with a couple of different species. And I was wondering, when you are considering what to remove, would you consider preserving as seed some of those redundant accessions?
Yeah. So really, when I speak about the redundant redundancies, right? We're just talking about sport cultivars, right? Not the wild species. You mentioned wild, right?
Yeah, for me, for the rubus, I have a lot of wild species, not cultivated.
Yeah, yeah. So one thing that we do observe in apple is in some of the wild apple is apple mixus, where the apple will actually clone itself, right, as seed, which is really useful, right, for preservation. So some of the explorations that Phil Forsland did brought back, Malus hoopiensis from different sites, and they turned out to be all the same genotype, right? So now we don't need 20 trees of the identical genotype. We could preserve their seed or just a single clone, right, in the field. No, but more what I was talking about are these sport mutations, right, which are prevalent in the industry. So like, geez, I don't even have a number offhand to how many red delicious apples there are. Just different sports that are a little bit more red or a little bit, more interesting, or they have some gala that are early ripening gala, right? And so some of those, like, yeah, it's challenging because you can't just eliminate them because like some might be useful for breeders specifically. But definitely, so here for these guys, we can't preserve a seed because we lose the unique allelic combination, but these could be possibly preserved only in cryopreservation.
Thank you.
I was hoping one of our field crew here in Corvallis would ask about fire blight. Does anybody have some questions on fire blight for Ben?
You don't want it, trust me.
It's too late, Ben.
So one of the things I'll mention from, so we typically rely just on streptomycin, right? Which is a little bit foolish on our part, but we're now, starting copper applications and a product called Blossom Protect, which comes highly recommended from our extension specialists. So using a multiple chemical and then a timed approach. So fire blight is challenging because of the timing, right? You time it when the trees are at a certain developmental stage. Well, we have tremendous genetic diversity for flower development and phenological development in apples. So there's no way. So if you spray at a certain time, you're going to be missing a good percentage of your collection. That's one of the biggest challenges is nothing we do fits the standard horticultural mold for preservation.
So Barb, you want to ask your question out loud?
Ben,
Yes.
let's see. We've been using Kasuman and Coside 3000, but because we cut our trees back, we have decided we're going to have to spray all summer long to protect the young shoots that are coming up.
Yes.
Because we're getting shoot infection even when there's no blossoms.
Mm-hmm.
Are you seeing that in apples?
Yes, we are, right? You see these pockets of infection that just kind of spread and spread. One thing that we've been poor at is really ideal timing of apogee applications, right? To really slow the growth down, which seems to slow down the infection rate. I don't know if you're also applying apogee.
No, we hadn't applied that. For the most part, our trees don't grow that much. Or how much growth are you seeing on your apples?
So we use we're currently on an MLA7 rootstock, which is a semi-dwarf, which so from standard seedling, it's about 60 to 70%, maybe 50 to 70% of what standard seedling is. But what we try to balance the heavy growth, right? So we have some trees on a dwarfing rootstock, which are just tiny, minimal growth, but then there's not enough to fill our demand for scion distribution. So we do have, so we're trying to balance that out. So we'll have, some accessions will put out a foot or more of new growth.
Right. So we were going to start applying once every week.
Yeah.
How often do you guys apply?
It seems about that. Yeah.
And when do you finally feel you can quit in the summer?
Oh, geez. Yeah. I think that's the hardest part in communicating with our field team, right? So again, the challenge is like the horticultural practices don't always apply to what we're trying to do as a clonal gene bank, right? So you'll have some people that want at the first sign of disease are ready to go chainsaw a tree and completely remove the genotype, which you could do in a commercial setting, right? You could yank out a tree that's infected, but we need every amount of budwood to propagate disease in trees. So last year was our particular challenge, but this year we're hoping to have a better control of the fire blight chemical applications, right? Not just rely. So we typically rely also on Cornell field services. This year, our field team has opted to take over that entirely. But I mean, we are spraying for fire blight with copper applications well into July and not quite August, but.
Oh, I have a question from Jane, who's also in my office. How do you back up your trees? Do you do depots?
What is that?
Do they have inside backup?
Do you have inside backup of your trees?
No, sorry. So no. So one accession of the Malus dumeri, we do have backup in greenhouse, but typically we do not. And that's something so we lose certain genotypes every year to fire blight, no matter if it's an outbreak or not. And that's something we're considering, just keeping them in a greenhouse. Unfortunately, as part of our fire 2 years ago, we lost our greenhouses, and we're kind of sheltering in place in our sister sites greenhouses for now, which are not nearly adequate enough for what we would hope to do.
Okay. Well, sometimes it sounds like sometimes you feel just like we feel we have to cut the tree down anyway. We try to back it up, but we do try to when they get that systemic infection down through their trunk, pretty much, I think they have to come out.
Yeah.
Don't you agree?
No, I 100% agree with that. It's a matter of cutting it down before you get the budwood, right? Before you're able to propagate it. So some of our some of the trees that were so heavily pruned, right? Let's see if I can find that. So some of these trees, right? Like you're not leaving very much for not only the tree to support itself and maybe outgrow some of the infection, but now you have minimal opportunity to repropagate that tree, which again, you're risking, keeping the infected tree around a little bit longer, but you're also, we need to balance that rescue of genotypes. Because some of these are the only one in the world potentially.
I know.
And that's where we do rely heavily on our cryo system, right? Gail Volk and her team are amazing at what they do. And it's without I think one of our crop I think is one of the highest backup rates across. I could be wrong. Gail will know, and she can share those details in her talk. But no, so that's where we do say this is not just end of the world. Our routine maintenance relies on rescuing these genotypes. So I guess I would say some of our trees we do keep, during the nursery stages, they are a little bit more protected, and that could be what we do for backup. So typically we'll do 3 to 5 trees of an accession, and then, right, so they're set aside from the main collection where they can get disease easily.
So are your surrounding trees, obviously you're in a situation where you have surrounding neighbors. Do you think they're infecting your trees also?
I think that's the hard part is, so Cornell is such a big place for apple production, apple research, but we really are the only apple collection on site on our particular farm, right? There's a neighboring farm maybe a mile away that has apple trees and other programs. So it's a challenge for all of us, right? So there's the apple breeding program, which maintains genetic diversity just on site, right? It's distinct from our collection. So it's a challenge for them as well. So we're sharing disease with our growers and with other research programs for sure.
And no wild or volunteers in the neighborhood, such as volunteer apples or volunteer pears or anything like that?
No, not like that.
Okay.
So the thing we're exploring now is adapting new rootstocks to better manage the disease. Again, it's not as straightforward as you put a resistant rootstock on a susceptible side and you get remarkable resistance, right? But just the way that rootstocks can mediate plant growth, and even the phenological timing can be somewhat modified with the rootstock. We're hoping that some of the more robust rootstocks can help protect our collections better. But what actually happened 20 years ago, which is fascinating, is that we started our collection in the 80s on EMLA 9 rootstocks or M9 rootstock. And then because of so many fire blight outbreaks, they finally switched to EMLA 7. So now 20 years later, right, we're looking at, let's look at new rootstocks and repropagate the entire collection. Only now we're a little bit more limited on space to do that.
Yes, I should say, you have so many apples. That's a lot of space.
So I was wondering, there might be some other questions from the whole group because we have many different represented. How about Hawaii or Davis or other sites? Any questions for Ben?
And I'm just new, so you guys can pick on me. So I'm still learning so much about everything.
No one's jumping up. Ben, I'll put in an intermediate question so that somebody else can think. How, especially with say grapes, well, with all of them, what is your procedure for pathogen testing? Are you making sure that the material you distribute is tested for viruses? And I guess that would apply to any of the cherries, the grapes, the apples.
Yeah, that's actually been a big kind of so interestingly, Geneva used to do all of its own disease testing and cryo like on site, and somehow along the line like that those programs died off and there hasn't been systemic testing. So we've currently been working with some virologists in Beltsville to test our cherry collection, and then we did a big chunk of our apple collection just looking at viruses. But unfortunately, that has not been a very active program, in our recent history. So it's definitely something to consider as, we just want to be good stewards, right? So we opted because of our fire blight outbreak to not distribute because we knew the particular strain was streptomycin resistant, and none of our colleagues should have to deal with that, right? That's a frustration to the apple community that we're trying to serve. Similarly, we don't want to impact, our growers or researchers or frustrate their programs by sending them diseased materials. But it's a challenging pill to swallow because it's either don't distribute anything because it may have disease and then no one gets the research material that they want, or like how do you systematically regenerate your collection in disease free or have it certified and then keep it certified disease free, right? Like when you have just so much genetic diversity and so much else to manage, like how do you balance that disease aspect, right? Fire blight's different. Like if you don't do something, it's going to melt down. So certain apple latent viruses, right, just accumulate and seemingly have no effect until all of a sudden they do, and then it can be devastating. These are the things that keep curators up at night, right?
Yes, absolutely. They're keeping me up too. With your grapes, is it the same way?
It is, yes, the same way. So more recently, I think Cornell virologists had tested the material in Geneva. So we don't have some viruses, right? But again, it's in a state of flux constantly. So every several years, it would require new evaluation and new consideration. That actually, as we get into looking at new rootstocks, some of the requirement for a lot of the nursery sources, right, are disease-free materials. So now you're hitting a wall with like, okay, we have new rootstocks we want to try, but if we can't get disease-free material, then there's an incompatibility issue that could arise. So so now it comes back to by our maintenance, not only our distribution.
Ben just put in a question, Ben Karstens. He says, Jeff Karstens. He says, Ben, nice job. If the Malice collection maintains 3 to 5 clones per accession, would there be a benefit to utilizing numerous rootstocks as opposing to switching the entire collection to a single rootstock?
Yeah. No, I didn't really mention those details in the collection. So typically we'll start with 2 trees on EMLA 7 and then cut out the odd numbered one or the even number one, or really actually depending on which one looks healthier at the time, but typically it's the even number to really thin out the collection. We've discussed now after the fire blight incidents, like, do we just keep both trees and just prune them smaller? Ideally, right, like I would love to keep 3 to 5, 7 clones of everything and just have a beautiful replicated block. I know our researchers really want that because when they go in to evaluate material and the trees are side by side, they don't like that you can't eliminate the environmental variation. But then now we're maybe 80% capacity for field on our field site for apple. So adding additional clones, right? Like who gets cloned and how many do you need? And I actually think Gail's program did a lot of exciting things looking at seed diversity. So potentially some of our seed diversity that's, or seed accessions that are, conserved as alleles, not as clones, right? Could help, maybe thin out some of the collection. But yeah, that's another question that we kind of wonder a lot about. So a lot of the apple production is going into high density. So there's some push to get a high density apple collection, and we could have 7 trees, right, on really closely planted together, high density setting. But then there's trellis and other things to consider. But then also like the budwood, like some accessions are so popular every year that we would strip a small tree, let alone 3 or 4 smaller trees of all of their first year wood potentially. So I mean, so certainly something, yeah, that's a wonderful consideration, Jeff. And I just can't figure out how to implement that, right? That best serves, the maintenance and our stakeholders.
Jeff, did you have a follow-up question, or is that good? Okay, he says good.
Good, thanks.
Ben,
this is Jenny.
Oh, good.
Sorry, Kim. I don't mean to talk over you. So at this point, do you only maintain a single clone of each accession in the field? Is that correct?
Correct for apple. So we do keep 2 to 4 vines of our grape collection because those are a little bit more volatile than the apple. Again, that culture, that mentality might change after this last year, and we had such bad disease. And then the tart cherries are kept as 2. I think the idea is that we had this wonderful safety net with cryo, right? And that we could just, we'll just eliminate 1 tree and propagate it again. Well, some of them were hitting that there's only 1 tube left in cryopreservation, which means like you've got to have a really good reason for pulling that last sample out and using it. And so some of them, you're like, okay, we can't, it's out of the collection, not quite permanently, it's still an accession, but now it's only maintained in cryo.
Sure, that makes a lot of sense. We have some similar challenges. We've got some accessions where we've got 2 clones in the field, and then there's others where we just have 1, and some of it's because of space, and some of it's because they're difficult to propagate or one has died. So we face a very similar challenge of who gets multiple, and sometimes it'd be nice if there was more, but land gets into the way. So I completely feel you on that.
That's why I love our tart cherry collection, because you can actually get your hands around it, right? It's small enough to be manageable. When you have like even our grape collection, like the Davis is twice as big as ours. Like, how do you know which genotypes are priorities? So certain things have died, and some researchers have said like this was the most important thing in my research, right? And that's really devastating to hear as a curator. But you're chasing so many things. If only stakeholders could provide like exactly what are the top priorities, because again, it's that eternal balance of like we want as much genetic diversity as possible, but then we also like have to maintain what we already have. So we don't want to like limit our new growth by planting too many reps, but then again, we don't want to lose the trees that we have already. And so Jeff has another question about single clones and how do we know when to cut off the deadline? So we do all of our own pruning and other things. And so again, we have that nice dormant cycle. So we just cut off every winter distribution. The deadline is October 1st, which may seem maybe early. I don't know what other sites do, but so that's the deadline to get in the request. The material is collected in December and is shipped in January. And then we have a summer request that ends in July, that then they can request summer budwood for apples and tart cherry. And it is weird how it's like, I don't know, it's like the stakeholders talk amongst themselves and decide like, we all want this apple. And then you'll see like 200 orders come in and somehow like of all the thousands of things to choose from, they all choose one or two of the same. It's I don't know. I would love to just pick their brains and get more feedback from our stakeholders and just see how they really operate.
We, just as a comment, we too see that there's trends in germplasm requests, and someone will put out on a garden web a blog that white strawberries are in. And so then we get a whole mess of requests for white strawberries, or, you know, like you say, for one particular clone gets written up and then you get a lot of requests for that. So
Yeah.
there was one other comment from Gail about the CGC having input in replication and suggestions and stuff like that. So probably as you work with your crop germplasm committee, you'll have lots of opinions and help on making decisions of how many things to be planting.
I love crop germplasms, the committees. Like you never even have to like think of ideas. They'll just give them all to you. Like, they'll even debate very heavily over what you should be doing as curator. And that's kind of a fun position to be in sometimes.
Yeah, definitely. And it sounds like Jenny Smith is saying that Davis has that run on a particular accessions clones too. So it's nice to know that we're in misery all together here, I suppose.
So when we looked at our top requested apple cultivars, they were almost all the ones you could get at Lowe's. Kind of like, that's kind of discouraging. Like, we've got the elite collection of such, diverse apples. Why do you want Golden Delicious? No, and I really appreciate this group and these trainings. Like, this is fantastic for me, as I still think of myself as new, but now like 3 years into the job, right? That's still new for anyone that works with perennial crops. And now we're getting a new Vitis curator, like having these as resources to learn about the culture and the dynamics of other clonal sites. Like, sometimes you forget that there are other ways of doing things.
Well, I think we've just about used up all our time, and that was an excellent presentation, Ben. I really appreciated your slide set and your example. Thanks very much.